Hi everyone, welcome back to another video. Today I'm going to be talking about DNA damage and repair and a little bit about mutations. I'm going to be talking about repair mechanisms. And yeah, that's pretty much it. I'm going to be popping out a few videos within the next couple days because I have a midterm on Tuesday and this helps me study. So yeah, let's get into it. So as always, I have a game plan. Today we're going to be covering introduction to mutations, what they are, and like kind of what causes them, and then DNA repair mechanisms. And that's pretty much it. It seems like it's a lot. Like it's only three things that are on the game plan, but there are a lot more mechanisms. The only mechanism I'm not going to cover today is recombination repair. I'm going to do that in a separate video because it is a lot. I literally had a separate lecture about it. So if it is a separate lecture, it's going to be a lot of stuff. So yeah, let's get into the first one. The first one, introduction to mutations. Just before that, I want to say if you hear any lawn mowing noises in the back, there's people doing yard work outside my house. So, anyways, back to mutations. What are they, you might ask? They are changes in DNA structure that can be good or bad. Good can mean like evolutionary wise to benefit um, one species or organisms, bad as in cancer, disease, death. So there's good and bad to mutations, or I guess if you're talking about DNA damage, it's always going to be bad, right? But those damages can cause mutations that can be good or bad for any organism, right? So the types of mutations that can occur are one, point mutations, which are just a single nucleotide substitution, small indels, which can be a small insertion or deletion, and then large scale mutations. I feel like that's self explanatory, like huge mutation gone wrong? Okay, so point mutations are base pair mismatches when not repaired in time. So basically, there's going to be a mismatch, and then replication will occur. And when that replication occurs, there's going to be a mutated base, and you're going to want to get rid of it or change it. Um, so yeah, that's pretty much we're going to talk about repairing those bases. But in the meantime, we're going to talk about point mutations. So an example of this will be a deamination from cytosine to uracil. So cytosine has an amino group, an NH2 group, and that cytosine will be deaminated. And so that amino group will be changed to a carboxyl group. Once changed to a carboxyl, the uracil is only able to form two hydrogen bonds. And so when you are doing like transcription and translation, all that type of stuff, your uracil is your thymine, quote unquote. So thymine bonds with adenine, right? And so when cytosine cannot bond with guanine, you're not going to have those three hydrogen bonds because you just changed the amino group to a carboxyl group. And so after DNA replication, uracil has to bond with adenine. Like there's no choice because uracil cannot bond with guanine. It doesn't have room to make a third hydrogen bond. Like it doesn't work like that. So because when you do replication, there are two strands. So one strand will have that mutated base of uracil and adenine, and then the other strand will have cytosine and guanine, which is good because you do have, at the end of the day, you do have one strand that is good, and then you do have one mutated strand. But at the end of the day, you do have a mutated strand of uracil and adenine, and it's just going to be like a what? Because you're doing DNA, you're not doing RNA, right? So, like I said, after that first round of replication, without fixing the mistake. So if DNA polymerase and ligase don't come in and add new nucleotides after that base has been excised, then you're going to end up with a mutated strand and a regular strand, right? So, yeah, pretty much you do have a good strand and a bad strand. But this is a transition mutation because you're going from a pyrimidine to a pyrimidine. If you're going from a pyrimidine to a purine, that is known as a transversion. So you can go from like A to C, and that will be weird, you know? Um, so yeah, that one is what I wanted to cover is that there are transitions which are pyrimidine to pyrimidine or purine to purine, and then transversions are like pyrimidine to purine or purine to pyrimidine. So at the end of the day, that's what you're gonna get if you do have a switch between those two. So with that being said, there are consequences to point mutations. Out of there's like three types of consequences. One of them will be a missense mutation. So it replaces another amino acid for It will just replace another amino acid. So instead of, let's say, tyrosine, which is UAC, you can replace it to serine, which is UCC. So instead of the adenine originally being there with tyrosine, you're replacing the adenine with cytosine. So that right there is a transversion um, mutation because you are transitioning between a pyrimidine and a purine. Another consequence would be a nonsense one, which produces a premature stop codon. So instead of UAC for tyrosine, you get UAA. So the last that last amino acid right there, instead of getting adenine, or instead of getting cytosine, you get adenine, right? And so I like to think of it as nonsense. Is like, if somebody's talking nonsense to you, you don't want to hear it, so you just want to stop hearing their nonsense. So I guess if that's a way to help you remember it, then I guess think of it like nonsense is stop codon because you don't want to hear anybody talk about nonsense, right? Like, just stop talking. So the last one is a silent mutation consequence. So basically, so when it's silent, it just means that you don't hear anything, as in you're not going to see any change. You're not going to hear any change in the you won't hear it, but you won't see any change in the protein. So instead of UAC, you get a UAU, and tyrosine is also UAU, because there's different. If you have seen a coding table before, you have seen that there are different codon sequences for the same protein. So yeah, that is point mutations. Another type of mutation are small indel mutations. So there's a section of like frame shift. So if you add a, nucleotide at the 5' end, you're going to disrupt the entire protein. Because when you do that, you're adding something at the beginning. So if something goes wrong at the beginning of your day, that might affect you for the rest of the day. If you're adding something at the 3' end, let's say you had something bad happen to you at the end of the day, and the rest of your day is good, then you're just only going to think about the last part, right? If something happens to you at the beginning of the day, let's say you miss your bus, then you might be missing like, let's say you have an exam that day, you miss your bus, you're going to be late to your exam, then you're cooked, right? You don't know what you're going to do, because your whole day is ruined. You missed your exam, which means you probably failed your test, which means you probably flunked the semester, right? So if you had something bad happen to you at the end of the day, let's say maybe, I don't know, what to say for the end of the day. If you work at a pizza place like me, and let's say you burnt a pizza at the end of your shift, then that's kind of sad. It sucks. But I mean, for this part, it's like the rest of your day was good. You just burnt that one pizza. Another thing would be in frame, so that would be codons, like three multiples. You can insert, delete, and then put a stop codon. Like, it's not much to explain for the in frame stuff. If you insert codons, then you insert, and if you delete, you delete. Also, I don't know why I'm eating pizza and doing homework at 2 a.m. in the background, but here I am. So enjoy this time lapse of me. Next thing we're going to go into is causes of mutations. So causes of mutations can be one, deamination, like we talked about earlier with that point mutation. So this can occur by spontaneous hydrolysis. So a 3' hydroxyl, which is literally our biggest op, literally will come in and steal your girl. Like, there's nothing to it. It's just because of the fact that 3' hydroxyl is super reactive, right? And so it'll see all these bonds, and it's like, I want them, right? So it's going to spontaneously hydrolyze, and through that you're going to convert your amino group to a carboxyl group or a hydroxyl group. And yeah, so let's just think of that from point mutation, and I think that is beneficial to think about, because at the end of the day, that this does happen quite a lot in our body. Another thing about deamination is that thymine cannot be deaminated because it doesn't have an amino group. So I would um, be wary of that as well if it comes up on an exam for anybody. Thymine does not have an amino group, so it can't be deaminated. So if your professor says true or false, thymine is deaminated by this route, you're going to say no because it doesn't have an amino group. Um, Another cause of mutation will be depurination slash depyrimidation. So that base is removed. So cytosine will be removed, but you're still going to have your D, so your deoxy. You're just not going to have your nucleotide. So DNTP, you're going to get rid of that D, or you're going to get rid of that N, and then you're going to keep your P. So it's DP. Oh wait. Anyway, so then next we have oxidative damage. And so oxidation, or all four nucleotides can be oxidized, so they're going to be mutated because they're not going to be, your cytosine is not going to be a cytosine after it's oxidized. Your guanine can become O6 ethyl guanine. Actually, is that an oxidation? I'm not sure. But at the end of the day, all four nucleotides can be oxidized, which does change their structure, right? And the last thing I want to talk about is alkylation. So alkylation adds an alkyl group to either the base or the phosphate backbone. Okay, so the most common areas to alkylate a nucleotide will be the N3 or nitrogen 3 of adenine and the oxygen 6 of guanine. So that's that carboxyl or that carbonyl. Have I been saying carboxyl this whole time? It's carbonyl oxygen or that carbonyl group. I'm so sorry. I meant to say carbonyl, not carboxyl, because carboxyl I think is totally different. Anyway, so those are the common sites of being alkylated. Okay. And so I have here on the screen is an example. So guanine and cytosine. Remember when I talked about point mutation, there was a deamination from cytosine to uracil, and then uracil formed with guanine. But uracil can't do that, so it formed with adenine instead. So you went from a GC to a UA or AT pretty much. But in this case, it's almost the same thing. But guanine gets alkylated at the O6, and you can see I have it in purple. I don't know if you can tell, but it's in purple. It's a CH3 CH3. Sorry, that should be CH2 and CH3, not CH3 CH3. But ethyl guanine loses a hydrogen bond pairing. Okay, so it cannot form with cytosine because cytosine forms three bonds with three hydrogen bonds with guanine. So because there's a lost hydrogen bond, ethyl guanine is going to bond with thymine instead. Shocking alert, right? So you're going to have two bonds, two hydrogen bonds with ethyl guanine and thymine. So the hydrogen bond was displaced, carbonyl oxygen cannot hydrogen bond, and guanine cannot do the three bonds with cytosine. So at this point, you basically become a quote unquote AT pairing. So you go from a GC to an AT. You go from a guanine cytosine to adenine thymine, even though your adenine is not adenine, it's ethyl guanine. But because of the fact that they form two hydrogen bonds. So you are doing a weird conversion at this point. Okay, so you can actually reverse this. And you're going to use this with an alkyl transferase. This is just going to transfer that alkyl group somewhere else for reasons that I don't know where, but um, it'll basically be gone. And this is a dealkylation. So this is done to, I think by doing this, then you're restoring your GC, which is what you want, right? It's a reversible reaction, like you can do this back and forth. But yeah, next up we're going to be talking about how to fix these mutations with repair mechanisms. Sorry, I actually lied. There is one thing I do want to talk about is radiation. So there are UV and gamma ray and X-ray radiations. So with UV radiations, it creates a cross-link between two adjacent pyrimidines on the same strand. And typically this happens with thymine-thymine, so you're going to have a dithymine dimer, and it disrupts the hydrogen bonds between bases and creates a kink in the DNA. So basically, you're going to have a linkage between thymine and thymine right beside each other, but then you're going to create a gap because adenine cannot bond with thymine. So you're going to have a big gap in your DNA, which creates a structural distortion. With gamma and X-ray, it creates a oxygen species or a reactive oxygen species that can break one or both strands of DNA, and you can repair that through recombination repair, which I'm going to talk about later. Okay, so one of the DNA repair mechanisms is called direct repair. This is when a damaged base is directly repaired on site and not excised out. This can be through dealkylation by alkyl transferases, as we just talked about. Pyrimidine dimers are repaired by photolyase, so this is a direct photoreactivation reaction, and that uses energy from light and breaks the covalent bonds by linking adjacent pyrimidines. But this one is pretty much, I don't have a photo or like a detailed mechanism for this, but it's pretty much just like it will just repair on site. It's like on site, you know? But yeah, next we're going to go into base excision repair. Okay, so for this mechanism, not to be confused with nucleotide excision repair, we're just taking out that base. Like, we're not taking out the full, like, length of nucleotides, it's just the one base. So as I'm saying here, at this point we have a strand. I just put in like random nucleotides here or bases, so it's nothing really crazy. You have a thymine and adenine, 2 hydrogen bonds, guanine, cytosine, 3 hydrogen bonds, and then an adenine and thymine, 2 hydrogen bonds. Okay, and so the base in green, so our cytosine in green, is going to be deaminated. So we talked about this earlier where that cytosine, where it deaminates, is going to become a uracil because we're changing that amino group to a carbonyl, right? So cytosine is converted into uracil by deamination. Recall our point mutation where we just talked about that, right? So yeah, so now we have guanine and uracil. We can't do that, right? We can't form 3 hydrogen bonds with uracil. It's just going to form with adenine. So glycosylase is an enzyme that's going to locate our uracil and cleave our bond. So like I just showed, like it's going to cleave it. So if you look at that red line, that dash line there, it's going to cleave it there. And that's going to leave an AP site, or also known as a apurine apyrimidine site. Okay, or just AP, right? Because it can be apurine, apyrimidine, but I think in this case it's an apyrimidine. So now AP endonuclease. So our apyrimidine endonuclease is going to cut out and excise that section where our base was, right? And now we have a gap. So what are we going to do about it? We're going to take DNA polymerase 1 specifically and ligase. So with that gap here, we're going to fill it in with DNA polymerase. Okay? Or we cleaved out that AP site, right? And so DNA polymerase will fill in the gap and we'll put in a new cytosine, and ligase is going to ligate it closed. And that is pretty much base excision repair. And so here's the big picture if you needed a better image. But yeah, I'm going to move on to nucleotide excision repair. Okay, so nucleotide excision repair, also known as NER or NER, NER? It's nerve. Okay, so this kink in the DNA is a thymine-thymine dimer, because of the fact there's no hydrogen bond in between that peak in the red that I put on the video. So there's hydrogen bonds on either side, which means adenine cannot form with thymine. So how are we going to fix this? We're going to take two UVRA molecules, and they're going to form a dimer. That is going to be combined with UVRB, and together they're going to bind to the lesion. This step requires ATP hydrolysis, so ATP goes to ADP plus phosphate, or a phosphoryl group, and so they're going to scan the DNA, okay, and they're going to find that lesion. Once they find the problem, once they find the thymine dimers, UVRA is going to disassociate. It's going to say, bye-bye, I don't want to see you anymore, to UVRB. And they broke up for the 40,000th time again. And so when that happens, well, hopefully this video moves on to the next step before I lose my mind. Yes, UVRA dissociates. We've already established that. Okay. Anyway, so UVRB is going to melt the DNA. It's going to be kind of annoyed that UVRA broke up with them again. So UVRB is going to melt the DNA locally. So just around that area where the thymine dimers are, where that kink is, okay? When that happens, UVRC is going to come and bind and form another complex with UVRB. So UVRB just got a rebound, and that's UVRC. And so once that happens, UVRC is going to nick or cut the 3' end and the 5' end of the thymine dimer. So it's going to basically cut on either side of that lesion and not, anywhere else, because it's only going to be bound to there. When that happens, helicase or UVRD is going to come in, do its thing, and the whole sequence is going to come out. So that sequence that had that thymine dimer problem is going to come out, which leaves you with an open wound, or the heartbrokenness of UVRB. UVRB will also disassociate as well. So UVRB and UVRC will disassociate, and there's going to be an empty sequence. Oh, I just did it wrong. I just rewrote it because that red is that thymine dimer, if you couldn't tell. Basically, that's what happens. So now DNA polymerase 1 and ligase are going to come in, do its thing. Like I said before, DNA polymerase will go in, put in some new nucleotides, and ligase is going to come in and say, we're done. And this is the full picture of the whole process. So the last repair mechanism I'm going to talk about is mismatch repair. So basically what happens is, as you can see, I have a replication fork if you can tell. And so DNA is not always going to be just flat, doing its thing. It's going to be methylated, it's going to have stuff to it. Like there's going to be methylations, alkylations, all that kind of stuff. So you're not going to see like a boring DNA. You're going to see some stuff going on. So what this mechanism does is that it finds the methylated DNA to fix the new DNA, because new DNA is not methylated. So it's like a hemimethylated thingy going on. I don't know if I wrote it down. But basically, in E. coli, the strand differentiation is based on DAM methylase. So it's going to look for the basically to methylate stuff. But yeah, so at this point, you have an unmethylated new strand that has a mismatched base. And so what we're going to do is we're going to recruit mute S and mute L. So they're going to make a complex and they're going to bind to the problem. They're going to bind to the mismatch. And so when they bind to the mismatch, they're going to scan the DNA bidirectionally. So I kind of put it as they're pulling the DNA. They're pulling the DNA to find the methylated parent strand. And that parent strand, for this case, I believe the sequence is GATC. That is the unmethylated strand, the new one. That's the nearest GATC site. That's where they're going to look for, or that mute SL complex is going to look for GATC. That is on the new strand. The old strand, I believe, starts with CTAG or whatever. Um, yes, I put that there. So the next step after this is once they have found that GATC site on the new strand, mute H is going to be recruited. And so mute H is going to do a cutting thing. It's going to cleave some stuff. I will show you as soon as I move the screen down. Thank you very much. Okay, so mute H is going to come in and it's going to bind to the GATC site and it's going to cleave GATC. Okay, it's going to cleave it. So when that happens, a special thing occurs, and your favorite friend helicase, but in this case it's helicase, I believe it's 2, helicase 2, will come in, and it's kind of shaped like a donut, low key, like the mochi donut. It's going to come in with exonuclease. So helicase is going to unwind the DNA, the new one, from where it was cut at GATC, that sequence. It's going to unwind that DNA, and exonuclease is going to come in and it's going to eat everything in the fridge. Okay. It's going to chomp away where that new strand was unwound, and it's going to eat up more than just where the mismatch was. It's going to go further than that. So it'll just eat up a little bit more than what it's supposed to. And because of that, DNA polymerase 3, in this case 3 and not 1, because I know in the other examples it was 1, DNA polymerase 3 is going to come in, add some new nucleotides, and ligase is going to ligate. And that's it. And here's the finished picture. Thank you guys for watching, and I'll see you guys in a couple of days actually.